Welcome to Lecture 9. Lecture 9 has several different topics. A big part of Lecture 9 is going to be the human microbiota. And so we'll talk a little bit about that, and we'll talk a little bit about the factors that control infections and infectious diseases. And there's going to be a lot of terminology that you need to discuss infectious diseases. So keep in mind when we're learning new terminology to make sure you're thinking about what it's defining. And if there are two words that are opposites of each other, think about that as showing you the range of possibilities between them. Whenever possible, try to take the terminology you see, and as you're going through these, practice using them. Yeah, so let's get into it. Where we're going to start is by thinking about symbiosis, because ultimately this is how we could describe any relationship we have with microorganisms. Whenever two organisms live close together, two different organisms, that's a symbiosis. And there are all different possible symbioses happen with us. For example, mutualism, that's when the host and the guest, the host is us and the guest is the microorganism. If we both benefit, that's a mutualism. And we could see this with things like some of the lactic acid bacteria that live on us and in us that help protect us from other bacteria. The benefit we get from them is the protection. The benefit they get from us is food and a place to live. The opposite of that is parasitism, where there are a lot of different microorganisms that really like to grow on us and in us, and they damage us. So, for example, the bacteria that grow on teeth, the late colonizers of dental biofilms, that ferment sugars to make acids that damage tooth enamel and cause tooth decay. Those are damaging us. So they have a place to stay, they have food, and we suffer. That's parasitism. Something that you'll hear a lot if you read about the human microbiota or the human microbiome, those are two separate words for the same thing, is commensalism, and that's when the guest gets a place to stay, the guest gets food, the host doesn't notice. There's no effect on the host. This probably happens a lot in our gut with, well, yeah, it probably happens a lot, but we don't really know. It's hard to define something as commensal if you can't see whether it has a beneficial effect or a parasitic effect. Scientists have to kind of classify that as we don't know. We have to keep looking. And so that is actually the most common symbiosis we have with microorganisms, is when we know they benefit because they're living on us and growing, but we don't know what effect they have on us. That is the most common, and we don't have a word for this. But functionally, this is the most common symbiosis we're part of. So, as we look at the human microbiota, first, we're really going to be seeing a lot of mutualism and probably some commensalism and this unknown situation. So we'll get there in a minute. Some big terms that we'll use again and again include things like disease and infection. Disease is any situation where the normal state of health is changed for the worse. And you could define this as saying a body system isn't doing its normal function. There are lots of different ways to think about this, but disease generally is going to be when the health state is changed. Infection is any state where there's a microorganism growing in a person or on a person. So if it's able to grow and multiply, we call that an infection. So that doesn't necessarily mean disease, right? That could be one of the mutualists that that grows in our guts or on our skin. And they benefit us. So when you see infection, don't necessarily assume disease. That brings us to infectious disease, and that's when a disease is caused by an infection. There are a lot of terms related to pathology, or the study of disease. Pathogen is one I've used a lot. That's an organism that causes disease. And we could also talk about pathogenesis, pathogenicity. I won't use these very much, but if we talk about how a bacterium causes disease, we are talking about ultimately its pathogenesis. So these two, pathogen and pathogenesis, we're going to use a lot. Okay, so those are terms we will consider a lot. Now let's get into the human microbiota. The first thing is, why do we call it that? Microbiota versus microbiome. You might also see human microbiome. Human microbiome refers to the genetics, and the human microbiota refers to the entire collection of microorganisms. So this is the correct term, and it is slowly becoming more and more accepted. I'm going to go through this with these little codes up in the left side. These will hopefully help you keep track of what of how the slides are organized. So all the slides on the resident microbiota, the human microbiota, is also known as the resident or normal microbiota. They're RM and then a number for resident microbiota 1. So you'll see these historically called the normal flora. Flora refers to plants, and we're not talking about plants, so we don't use that term anymore. There are huge numbers of microorganisms in us. The vast majority of them are in the colon, but also they're present on any other site in the body, and there are 10 times as many of bacterial cells as human cells. So this isn't important by mass. Those cells are tiny. Bacteria are much smaller than human cells. But if we have a diverse array of microorganisms, they carry in their genomes lots of different genes that allow them to do metabolism that we can't do. They can make products like vitamins that we can't make in our cells. They let us digest a wider range of foods than we could do using our own enzymes. And so if we think about the microbiota as contributing genes that we ultimately indirectly use, they do become very important. Additionally, for every organism that exists on Earth, there is some kind of virus that infects it. And for every cell, there are 10 virions, roughly. So virions are more common than cells of any type. Anywhere in the world, in any environment, there are more virions than cells. Let's see. Right. So typically, we find microorganisms from our normal microbiota or resident microbiota on any external surface, so like skin, and then any mucous membrane. So the conjunctiva, the respiratory mucous membranes, the entire alimentary canal, those are all colonized. The male and female urethra, the vaginal lining, we find bacteria and other microorganisms in all of these places. And that's what we expect. Skin and mucous membranes all have adaptations to help control the normal microbiota that are found there, and kind of keep them in the right place. If we find microorganisms anywhere else, so like deep in a muscle, that's a disease state. Something is wrong in that situation. But if we find them on a mucous membrane or skin, that is normal. one thing we can make a distinction between is resident versus transient microbiota. This comes down to what are you doing when you are washing your hands, for example, or scrubbing your skin. We have a resident microbiota that are essentially permanently attached to our skin. From their point of view, that is permanent. And those are generally the less harmful ones. These are the microbiota. We also have transient microorganisms that fall on us or that we pick up when we touch something. And they don't have the same adaptations for sticking to us that the human microbiota have. So the transient microorganisms either don't want to be on us, they don't benefit from being on us, or they're more disease causing. And so they can, their presence on us would be temporary. And so one major way we would contrast these is that the resident microbes can't really be eliminated. We can't get rid of the resident microorganisms on a person. We can't sterilize the skin. But things like hand washing, scrubbing skin using iodine, those don't get rid of all the resident microbiota. Hand washing can't get rid of even a big fraction of the resident microbiota. They are tightly attached to receptor sites on our skin, and washing doesn't get rid of them. The transients, though, they're not tightly attached to us. And so when you wash your hands, you would remove most pathogens, you would remove most harmful organisms from your hands. This is a table that can give you more information about what's going on with the microbiota at different parts of the body. So this comments column is going to be the most interesting to you. And I put this CUR label up here again to signal that this is for your own curiosity, and I won't test you on the content of these words here, but I do expect you to know that all of these sites are colonized by bacteria, because these are all either external sites or mucous membranes. We get our microbiota during birth, and it stays with us all through life. The bacterial communities in our guts change very slowly during adulthood. They might change in appreciable ways over a scale of years. Whereas in a newborn, they're changing very fast because a newborn is not colonized until birth. So the fetal environment, it would be sterile. But during birth, the child would be exposed to bacteria either from, different bacteria depending on whether it's a vaginal birth or a cesarean. So cesarean birth colonizes the newborn with skin bacteria, and vaginal birth colonizes the newborn with the urogenital bacteria. And so the vaginal birth gives the newborn the microbiota they kind of need to set up for their gut, whereas the cesarean doesn't right away do that. And that's part of why people who are giving birth by cesarean section are now starting to intentionally expose the newborn to bacterial swabs of things like the vaginal microbiota, just to set up that early exposure to the microbiota. And then after that, a newborn would get very specific bacterial community during breastfeeding, because breast milk has really specific sugars in it that seem to be designed for feeding certain bacteria but not others. And so breast milk is going to kind of intentionally help control the newborn's microbiota. And then as the child grows and is exposed to adult food, some of the nutrients we find in adult food, things like dietary fiber, for example, those will support different microorganisms' growth in the gut. And so once a child transitions to eating adult type food, their microbiota will start to resemble an adult microbiota. So our microbiota have functions that we care about. They help us in several different ways. And one major thing we can expect our resident microbiota to do is to protect us from pathogens. And this is called colonization resistance, or the barrier effect. And this happens everywhere we have microbiota. So our skin, our respiratory epithelia, definitely in the gut, definitely in the vagina. We have lots of microorganisms growing there. And what we know is if something happens to them, we become more susceptible to diseases at those sites. So we know sort of indirectly that our microbiota protect us from pathogens, and in a few cases we know how it works. There are some cases where we know exactly what the bacteria are doing that are protecting us. And so that's a big thing the microbiota do for us, is protect us from pathogens. Another would be helping us harvest more energy from food. So bacteria and archaea in the gut can ferment carbohydrates that we can't digest. So dietary fiber goes through our alimentary canal without being digested or absorbed, but then bacteria in the colon and archaea in the colon can digest it, and then we absorb their waste products. So we can get some energy from food that we can't even digest because of them. The other really weird thing is that as we develop, as a human grows, the microbiota are something we sense. And things like our immune system, as it grows and develops, it needs the microbiota to give it certain signals. So we have data from other mammals like mice, where if we raise them in a sterile environment, which you can actually do for mice, you can remove their gut bacteria so that they are essentially sterile. Their immune systems don't develop correctly. So we are adapted to use whatever senses we have as we're developing. And in the case of the immune system, that's going to be the human microbiota. So how does the microbiota protect us, and what is it doing? There are a lot of different things we know about how our resident bacteria protect us from pathogens. And we can classify these into 3 different groups. One of them would be competition. So a good example of this is the bacteria on skin, our resident normal bacteria on skin, are very good at attaching to the available attachment sites. So proteins and sugars on our external epithelia that bacteria can attach to, the ones that are best at attaching to them are the ones that we want to attach to them. And so they can just compete for space and compete for attachment sites. In other situations, they directly attack each other. We have bacteria in the gut, and the gut and mostly the alimentary canal and the vaginal lining. We have lactic acid bacteria that make certain proteins that directly attack other bacteria. And so that's a specific antagonism. That's where we are expecting our microbiota to directly damage transient or dangerous bacteria. And then we have other situations where there's kind of an accidental damage that our microbiota do to invaders. So an example of this would be in the gut we might have bacteria that can grow on our fiber, and they make certain waste products. And those waste products would be toxic to certain pathogens and other bacteria that don't really belong there. And so just by virtue of doing what they do, they protect us. Okay, so that's enough for the first video. In the next one, we're going to look at dysfunction of the human microbiome and the human microbiota. We're going to see what can happen if it is damaged or altered. And then we will get into more investigation of how we understand infections and how we understand infectious disease.